Up until this point in the class, we've been talking about transmission genetics, how genes are transmitted from parents to the next generation and the subsequent generations. We're going to switch gears now. We're going to talk about genes in populations, and population genetics is what we refer to this as. And we'll talk about how we study genes in populations, their distribution, and how we can learn about the population and whether anything unusual is happening in the population using some quantitative parameters. And today's class, we're going to focus on two of the fundamental ideas of population genetics, and that is Hardy-Weinberg's law and Hardy-Weinberg's equilibrium. And so what I aim to do today is give you an intuitive sense of how we can use this sort of fundamental theorem of population genetics in order to be able to study populations and determine whether anything unexpected is happening in those populations. All right. Well, let's imagine a theoretical population, and I've drawn a population of little human beings on the board here. And so this is going to define our population here, all of these individuals. And in fact, we can formally define a population as a group of individuals of the same species. And when we're talking about population genetics, we talk about the gene pool. And the gene pool is the sum total of all the alleles in the breeding members of the population at a given time. And so let's look at this population and define some properties of this population. So the first property we can define is that there are n individuals, and in this case, n equals 16. There are 16 total individuals here. And what we're interested in here is locus A. And so we can talk about the number of alleles there are in the population for locus A. And of course, if you look here, you can see that there is big A and little a, and the total number of alleles is 2n equals 32 alleles in our population of 16 individuals. And of course, that's because we have a diploid population here. And so the question we'd like to ask, the first question we can ask is, what is the frequency of the different genotypes here? So remember that the genotype is defined by the two alleles. And so we can quantify those genotypes just by summing them. And so let's do that over here. So let's talk about how many of the big A genotype we have in the population. We just need to go through and count. There's 1, 2, 3, 4, and I see only, there's a fifth right there. And so we have 5 big A genotypes in the population. Let's do that for our other genotypes as well. So for the heterozygote, big A, little a, we have 8 of those. And for the homozygote, little a, we have 3 of those. And of course, now what we'd like to do is quantify the frequencies of those genotypes. So we just divide by the total number of genotypes in the population, which is the number of individuals. And so this is going to be 5 divided by 16, which is equal to 0.31. This is going to be 8 divided by 16, which is equal to 0.5. And 3 divided by 16 is equal to 0.19. And of course, if we add up the frequencies of those genotypes, they should sum to 1. And indeed, they do. All right, so we've counted up our genotypes and how frequently they occur in the population. And in fact, let's define those as frequencies here, just so we have the terminology. So this here is the frequency of our big A genotype. This is the frequency of our big A little genotype. And this is the frequency of our little a genotype. And so those are the frequencies of the genotypes. And what we would like to ask is, what is the frequency of each allele? And so let's figure out that then. So we'll leave the genotypes up there, and we'll ask over here, what is the frequency of the big A allele? And to do that, we need to look over here at our genotypes, and we see that the big A allele occurs in this genotype right here, and it occurs in 0.31% of individuals, so it's 0.31. But the big A allele also occurs in these individuals, in the heterozygotes, but it's present at half the frequency. And so in that case, it's 0.5, but we only want half of that. So it's a half times 0.5, and this is equal to 0.56 if you do the math. Now we want to ask, well, what's the frequency of the little a allele? So let's determine that, and we can write that here. And we can see that the little a allele appears in the little a homozygotes, and it occurs with a frequency of 0.19. But it also occurs in the heterozygotes, and it occurs, but only half of the alleles in the heterozygotes are the little a allele, and so we want half times the frequency of the heterozygote again, which is 0.5, and that is now equal to 0.44. And of course, 0.56 plus 0.44 is equal to 1. And so since we only have 2 alleles, that is as we would expect. And so we can generalize this now to the following formulas. So typically we talk about allele frequencies as being P and Q. And so in this case, typically we talk about the high frequency allele as being P. And as you can see in this formula, the high frequency, the P allele is present at the frequency of the big A genotype plus half the big A, little a heterozygous genotype. By contrast, the Q allele, so we typically use that by convention for the lower frequency allele, is present at the frequency equal to the little a genotype plus half the frequency of the heterozygotes. And as we saw in the example above, P plus Q is equal to 1. And they sum. And this becomes very useful because sometimes we just have one allele frequency. And of course, if we know P, then we know that Q is going to be equal to 1 minus P. And we can immediately compute the Q frequency once we know P. All right, so I've shown you how we go from our genotype frequencies to our allele frequencies in the population, which is what we're interested in this case. And I've written those up here again for you. So the frequency of the big A allele, which we typically use the symbol P for, is equal to 0.56. And the frequency of the little a allele, which we use the symbol Q for, is 0.44, and those sum to 1. And now what we want to do is think about the formation of the next generation of the population. So if we're looking at the current generation, we've computed these frequencies, and now we want to think about forming the next generation. And remember, as geneticists, when we're thinking about forming the next generation, whether it's we're thinking about 2 parents and the offspring, or now that we're thinking about populations, we want to be thinking about the gametes, that is the sperm and the eggs. And so I'll draw a little symbol here for the male gametes and for the female gametes. All right, and so let's think about now forming a random individual for the next generation. And to do that, we think about a process known as sampling the gene pool. So we randomly sample the gene pool in order to form a male gamete. And let's ask what would be the probability that that gamete would carry the big A allele. Well, of course, it is 0.56. So that's going to be P equals 0.56. And if that male gamete was to then fertilize a female gamete that was also sampled from the gene pool, and what would be the probability that it would carry the big A allele? Well, of course, that would be P equals 0.56. And so the probability that a child, let's draw the offspring over here, we'll keep them, the sex of them ambiguous. So the probability that that offspring is going to have a big A genotype is simply the product here equal to P squared. And that is, of course, 0.56 squared, which is equal to 0.3136. Now let's think about the little a allele. So if we randomly sample the gene pool to form a male gamete, what's the probability it's going to have a little a allele? Of course, that's going to be our allele frequency of Q, which is 0.44. And similarly, if we randomly sample and generate a female gamete, what's the probability it's going to have a little a allele? Well, that probability is going to be 0.44. And so when we think about the offspring, the probability that an offspring formed from the current generation is going to be homozygous for the little a allele is simply the product, which is Q squared, which is 0.44 squared, which is equal to 0.1936. All right. So those are the two cases in which we form a homozygote in the next generation. But we can also form heterozygotes by randomly sampling the gene pool, of course, by combining the big A allele with the little a allele. And so if we think about randomly sampling the gene pool and getting the big A allele, that's going to occur with probability 0.56. And if we generate a male gamete that has the big A allele, and then the probability that it will encounter a female gamete with the little a allele is equal to Q, so that is 0.44. And that's one way that we can generate a heterozygote in the next generation. But the other way is that if the male gamete carried the little a allele, which is going to occur with probability 0.44, and the female gamete carried the big A allele, which is going to occur with probability 0.56. So indeed, there are two ways to form a heterozygote. And therefore, when we do our computation here to figure out the probability of forming a heterozygote for the next generation, we sum those two together and get 2pq. And so the point here is that if we know the allele frequencies in the current generation, we can compute the expected genotype frequencies in the subsequent generation. And so, of course, those are going to be that the frequency of the big A genotype is going to occur with frequency p squared. The frequency of the little a genotype will occur with frequency q squared. And the frequency of the big A little a heterozygous genotype is going to occur with a probability of 2pq. And of course, these are all the possible genotypic combinations. And so if we added up those frequencies, they must equal 1. And so this is, in fact, the logic behind the following relationship, that if we add up all the frequencies, p squared plus 2pq plus q squared, it equals 1. And this is the famous formula known as the Hardy-Weinberg Law. All right, and this is actually a very simple mathematical relationship, and you can see that this must be the case. If you recall some of your high school mathematics, we started off knowing that p plus q is equal to 1, and so it follows that p plus q squared must be equal to 1 squared, which of course is 1. And if you expand this, of course, you get this. And so this all makes sense. All right, so this again is the Hardy-Weinberg law that describes the expected frequency of genotypes given allele frequencies. And so let's see how we can use the Hardy-Weinberg law. So firstly, what I just want to reiterate here is that this allows us to be able to do two things. Firstly, we can calculate, one, the allele frequencies in the current generation, knowing the genotype frequencies. So we saw that in our initial example. Similarly, we can calculate the genotype frequencies in the next generation, from the allele frequencies in the current generation. And this turns out to be a very powerful relationship. So let's take an example of how we could use that. So let's consider the Irish population. So in Ireland, there is a high frequency of the autosomal recessive disease cystic fibrosis. It's a disease we've encountered, and we know it's autosomal recessive. It occurs when you have two carrier parents. Okay, and the frequency, I will just erase this here. So the frequency of cystic fibrosis in the Irish population is, in the Irish, 3 out of 10,000 births is the frequency of cystic fibrosis. Okay, and so that is equal to a frequency of 0.03% of births the child is affected with cystic fibrosis. And so we know the genotype for cystic fibrosis, so we can figure out, we know this information here tells us what the frequency of the CFTR mutant, CFTR homozygous mutant genotype is in the Irish population is 0.0003. Of course, we have to divide by 100 to get here. So we've added 2 zeros, so that's correct. Okay, and in fact, now using this relationship up here, we know that this is our Q squared genotype frequency, and so let's put that here. So given that we know that the current genotype frequency in the Irish population is our Q squared, or 0.0003, we can ask, what is the allele frequency? What is the allele frequency? And to do that, what do we need to do? We need to determine Q, which of course we just take the square root of our Q squared. And so that's going to be the square root of 0.0003. And that turns out to be 0.01732 is the frequency of the cystic fibrosis disease allele in the Irish population, which we can compute knowing the genotype frequency, which we've just got from epidemiological data, knowing how frequently we get affected cystic fibrosis children in the Irish population. And of course, when we know Q, we know P. We get it for free. And so P is going to be equal to 1 minus Q. And so that's 1 minus 0.01732, which is equal to 0.983. And so P in this case is the wild type, or the non-disease causing cystic fibrosis or CFTR allele. Okay, and so we can ask a very important question here now. We can ask, what is the frequency of carriers of the cystic fibrosis disease allele in the Irish population? And so let's determine what that is. What is the frequency of carriers? Carriers, in this case, are the heterozygotes. And so, of course, looking over here at Hardy-Weinberg, we can see that to calculate the frequency, the expected frequency of the heterozygotes, it's simply 2pq. We calculated q over here and p, and we have those allele frequencies over here. So let's do that simple calculation. 2 times 0.01732 multiplied by 0.983. And if we do the math, that equals 0.034. So remember, this is our frequency of carriers. And the carriers in this case are the CFTR mutant, CFTR wild type individuals. And so this is our frequency. If we want to get it to a percent, we can multiply by 100%, which is equal to 3 0.4%. And so what this tells us, given the frequencies, the allele frequencies in the population, is that the frequency of carriers in the Irish population is 3.4%. And so this is an interesting illustration because the frequency of affected individuals is very low, 0.3%, but the frequency of carriers is actually very high, 3.4%. That's a large fraction of the population are carriers of that disease. And so this sort of illustrates the point that when an allele was rare in the population, most of those alleles, those rare alleles, are present in the heterozygotes. And so a problem that I'd like you now to work on at home by yourself is, given that we know the frequency of carriers in the Irish population, if you were going to be doing genetic counseling for some prospective parents and you had 2 Irish parents, what would be the probability that they would have an affected child? And that's something that you can compute directly from these numbers and your knowledge of Mendelian genetics. All right, so now let's ask how we can use Hardy-Weinberg to predict the genotype frequencies in the next generation. So let's think about our initial generation, which we'll call T0. And let's think about some genotype frequencies. So the frequency of the big A allele here is 0.64. The frequency of the heterozygote, big A, little a, is 0.32. And the frequency of the little a genotype is 0.04. And so we have our genotype frequencies. We can now compute the allele frequencies using the relationship that we learned. And so let's ask, what are the allele frequencies? And of course, the allele frequency of the big A allele is equal to the frequency of the big A homozygote plus half the frequency of the heterozygotes, which in this case is 0.64 plus half of 0.32, which is equal to 0.8, and that is our P. Now, of course, we know what Q must be, but let's go through the process to make sure that our mathematics is right here. So the frequency of the little a allele is equal to the frequency of the little a homozygote plus half the heterozygote frequency. That is 0.04 plus half of 0.32. And of course, that is equal to 0.2, which is our Q. And so P plus Q equals 1, and everything is good. Okay, so we have our current genotype frequencies. We can calculate the allele frequencies in the current generation. And what we'd like to ask is, what are the genotype frequencies going to be in the subsequent generation? Okay, and so to do that, of course, we can use Hardy-Weinberg's law up here. And so let's compute that for the subsequent generation. So let's just take our P over here is 0.8, Q is equal to 0.2, and let me just erase this calculation here, because now what we are interested in is what are the expected genotype frequencies in T1 in the next generation, given that these are the current allele frequencies. And so here's where we can use Hardy-Weinberg's law to be able to compute these values. So the frequency, the expected frequency of the big A genotype is simply going to be our P squared. And so that is 0.8 squared, which is equal to 0.64. The frequency of the little a is simply going to be Q squared from here in Hardy-Weinberg's law, which is equal to 0.2 squared, which equals 0.04. And then our heterozygotes, big A little a are going to occur with this frequency 2pq, which is 2 times 0.8 times 0.2, which equals 0.32. So here is T1. All right, so we started with these genotype frequencies in our existing generation, which we call T0. We have these frequencies of each of the 3 different genotypes. We use that to compute the allele frequencies. And now we've used Hardy-Weinberg's law to compute what is the expected genotype frequencies in the subsequent generation, the next generation. And what we see is that in the next generation, the frequency of the big A homozygote is 0.64, which is actually exactly what it was in the initial generation. The frequency of the little a homozygote is 0.04, which is exactly what it was in the current generation. And the heterozygote big A little a is 0.32, which is exactly what it was in the current generation. And why is this the case? Because this population is literally in equilibrium. And so this is what we define Hardy-Weinberg equilibrium as being, is when the population is literally in equilibrium, such as we observe with this population now. And so this was a really important finding when, even though the mathematics is, incredibly trivial, and in fact, these were mathematicians who discovered this principle and thought it was so trivial that it took them a long time to publish this finding. But the important implication here is that genetic variation is neither created nor destroyed by the process of transmitting from one generation to the next. And so the population, genotype frequencies remain stable in a population that is in Hardy-Weinberg equilibrium. And so just to convince yourself of that, I'd like you now to just work through the calculation and tell me what the genotype frequencies are going to be in the second generation, given that these are the genotype frequencies in the first generation, following the one that we are currently studying. So now that we know this sort of fundamental principle by which we can generate an expectation of the genotype frequencies in a population, given the allele frequencies, and vice versa, we can use that to ask the question, is a population in Hardy-Weinberg equilibrium, which is sort of like our null hypothesis. And so let's consider an example here. So this particular case is dealing with a HLA gene. That's the human leukocyte antigen gene, HLA-DQ1. This is part of the MHC complex, which is a very important in the human immune system. And so we're just going to think about a single SNP, a single nucleotide that varies in the population, and we have either an A or a G. And so we can do a very simple study where we can genotype all the individuals in that population and determine what their genotypes are. And we have a total of 17 big A or AA individuals. So this is now a SNP that we're talking about. We have 12 individuals that are homozygous for the G allele, so they're GG. And we have 55 heterozygotes who have the AG genotype. And so we have a total of 84 individuals that have been genotyped here. And so we can, of course, calculate now the frequency of each of those genotypes. And so that's very trivial over here. So this frequency is going to be 17 divided by 84, which is equal to 0.202. This frequency is going to be 55 divided by 84. That is equal to 0.655. And the frequency here is going to be 12 divided by 84. That's equal to 0.143. And whenever you're doing these sort of calculations, you always just want to stick this in the calculator and make sure, of course, that the frequencies add up to 1 so that we know that everything is good. So we have our genotype frequencies here. Now let's go and determine our allele frequencies. So we want to compute the P, which is the frequency of the big A allele, which of course is the frequency of the heterozygotes plus half the frequency of, oops, I said that wrong. This is the frequency of the homozygotes plus half the frequency of the heterozygotes. That's going to be 0.202 from here, plus half of 0.655, which is equal to 0.5295. And just to make sure I'm not doing anything weird, we'll compute Q directly from here. So that's this frequency plus half the frequency of the heterozygotes. So that's a mistake here. We want this genotype, and so that's this 1.143 from here plus half the hets, which is 0.655, and that is equal to 0.4705. And if we add P plus Q, it equals 1, and everything is as we expect. Okay, and so what we've done here is that we've taken the genotype, the number of individuals with each genotype, we've computed the frequency of each of those genotypes, and we've used that information to compute P and Q, the allele frequency of A and the allele frequency of G in this case. And so that's those are the frequencies that we observe in the population, but we can ask the question, what is the expected frequency? What is the expected frequency under Hardy-Weinberg equilibrium? Okay, and so of course you remember the formula for Hardy-Weinberg equilibrium, P squared plus 2PQ plus Q squared equals 1. And so now let's go and compute the expected genotype frequencies given these allele frequencies right here. And so for the big A genotype, what do we expect? We expect to see it, let me just keep out of the way of here, we expect to see that genotype frequency with a frequency equal to P squared, which is equal to 0.28033702. We expect to see the heterozygote with a frequency equal to 2PQ, which is equal to 0.4982595. And we expect to see the GG genotype frequency with a frequency equal to Q squared, which is Q over here is 0.4705 squared, and so that is going to be 0.2213702. And so we can already see here that these expected genotype frequencies, as computed on the basis of Hardy-Weinberg equilibrium, differ somewhat from these observed frequencies as shown over here. And so we can now ask the question, well, actually, how many individuals would we expect given that we have sampled a total of 84 individuals? And to do that, we'll just multiply this by the number of individuals, which is 84 for each of these. And this is our equal to 23.5511. This is equal to 41.8538 individuals. This is equal to 18.59510 individuals. And if we sum up the total of the expected number of individuals, that of course is going to be equal to 84. And so, and these, of course, we could do this check as well here and make sure that these genotype frequencies add up to 1. And so now we have a scenario in which we have the expected number of individuals under Hardy-Weinberg equilibrium, and we have over here the observed number of individuals. And we want to test the null hypothesis that this population is in Hardy-Weinberg equilibrium. And so you might already be thinking, how are we going to do that? And the big clue here should be that if we have the observed number of individuals and the expected number of individuals, we are of course going to use the chi-square test statistic. And so I'd like you to try and compute that now for yourself, remembering the formula for the chi-square test statistic. And you can use that to ask, is this population in Hardy-Weinberg equilibrium, or is this variation that we see, this deviation between the expected and the observed number of individuals, just due to the fact that we're sampling a relatively small number of individuals? All right, so here I've worked through that calculation. So of course, as I said, we are going to use the chi-square test statistic, and here is the formula written over here again, in case you've forgotten it, which hopefully you will remember it for the rest of your life. And so we have the observed value for each of the genotypes here, 17, 55, 12, which sum up to 84 individuals. And then over here, for each of those genotypes, we have the expected value that we have computed using the Hardy-Weinberg equilibrium relationship. And we're dividing, of course, by the expected value. And then we have computed, when we square them and divide by the expected value, we've computed the chi-square value for each of those three genotypic classes over here. And of course, what we want to do now is sum up those chi-square test statistics. So if we take the sum of all the chi-square test statistics, that's equal to 8.282. Now let's think about what we're testing here now. Our expectation, remember, for the null hypothesis is that nothing unusual is going on. And Hardy-Weinberg equilibrium gives us an expectation of what the condition in which nothing is going on, what things should look like. And so in this case, we define our null hypothesis as the population is in Hardy-Weinberg equilibrium, and the alternative hypothesis is that the population is not in Hardy-Weinberg equilibrium. And that's what we'd like to test. So now we have to determine, given that we've computed the chi-square test statistic for these genotype counts that we see in this population, the sample of the population, how surprised are we to get a test statistic that large or larger, given that the null hypothesis is true? And so here's our chi-square test statistic. But in order to be able to interpret that, of course, we need to know the number of degrees of freedom. So what are the degrees of freedom in this case? Well, this is just like when we're looking at the different phenotypic classes in Mendel's experiments. We know that in this case, we have 3 different classes. Okay, so these are our 3 different genotype categories. These 1, 2, 3. And so typically, as we saw with Mendel's phenotypic proportions, or in general with the chi-square test statistic, the number of degrees of freedom is the number of different classes or categories minus 1. In this case, however, we lose 2 degrees of freedom, and we only have 1 degree of freedom left. And that is because we've lost 1 because of the requirement that the number add up to 84, and so we lose 1 degree of freedom from that. And the other place in which we've lost 1 degree to freedom, so we have n, so we lose that one, but also we computed p from the data, and so we lost another degree of freedom. We actually got q for free, because once we know p, we get q, of course, because that's just 1 minus p. So in this case, we've lost these 2 degrees of freedom, and so the number of degrees of freedom in this particular chi-square test statistic is equal to 1. And so we, of course, now would go and plug that into R, and you should plug that in and find out what the p-value is. I will tell you that in this case, the p-value is equal to 0.004. And so we could say that we're going to reject the null hypothesis given this chi-square test statistic, because we only have a probability of 0.004 of getting a chi-square test statistic that large or larger when the null hypothesis is true. And so that's a pretty low probability. And so we have support for rejecting the null hypothesis. And so you can see in this case that, in fact, what we could also, if we investigate further, we could say, well, in fact, what we seem to have is an excess of this particular class. We have 55 heterozygotes, where we only expect 41, 42 heterozygotes. And so it's this particular genotypic class that is inflated compared to our compared to what we would expect from Hardy-Weinberg equilibrium. And that turns out to probably be due to a very interesting phenomenon that I'll talk to you about later in this lecture, in which we actually get preferential transmission or generation of heterozygotes in this population. Now I want to talk to you about how Hardy-Weinberg's law generalizes to multiple alleles. So of course, if you consider a single nucleotide polymorphism, there's 4 possible alleles. You could either have an A, a C, a T, or a G at any given locus in the genome. And so we can define those allele frequencies here as the frequency of the A allele is going to be equal to P1, the frequency of the T allele is going to be equal to P2, the C allele has frequency P3, and the G allele has frequency P4. And so we've just generalized our definition of allele frequencies. Rather than using P and Q, we're going to use P subscript 1 through 4, in this case for these 4 different alleles. And now what we'd like to be able to do is use Hardy-Weinberg's law to be able to compute the expected frequency of the genotypes on the basis of these allele frequencies that we've determined. And to do that, we use exactly the same principle, in which the frequency of the AA genotype is going to be equal to the frequency of our A allele, P1 squared. Similarly, the TT genotype will be P2 squared. The CC genotype will be P3 squared. And the GG genotype is P4 squared. And so those are just the allele frequencies for each of those individual alleles squared when we're computing the expected frequency of the homozygous genotypes. Similarly, as with the 2 allele case, when we're computing the frequency of the heterozygotes, we simply consider the frequency of each of the individual alleles. So for the AT genotype, it's going to be P1 multiplied by P2. And as we saw in the 2 allele locus, there are 2 ways to generate that genotype, and so we multiply it by 2. Similarly, the AC locus is going to be P1 multiplied by P3, and there's 2 ways to do that. The TC genotype is going to be P2 multiplied by P3. There are 2 ways to do that. The AG locus is going to be P1 times P4. There's 2 ways to do that, and the same logic holds for the rest of the heterozygous genotypes here. And this is how the computing the expected genotype frequencies is going to generalize from Hardy-Weinberg's law for any number of alleles at a particular locus if we want to compute the expected genotype frequencies for the homozygous class and the heterozygous class. And what's important here now, of course, is that if we add up all those genotypes, since they are all the possible genotypes that can occur in the population. So let's do that now. I'll get rid of these allele frequencies here. And if we add all possible genotypes up, which is P1 plus P2 plus P3 plus P4 plus 2P1 P2 plus 2P1 P3 These are all the possible genotypes that can occur in the population. And so if we add up the sum of all their allele frequencies, then that must be equal to 1. And then we know that we've done everything correctly. And so that's how Hardy-Weinberg's law generalizes to a multi-allelic locus where we have more than 2 alleles. So one question that I think is illustrative of the importance of Hardy-Weinberg equilibrium is to ask, well, how long does it take for a population to achieve Hardy-Weinberg equilibrium? And so we can do this experiment just by simulation. So let's take our Italian population, in which we had 84 individuals, and we had demonstrated using the chi-square test statistic that they were indeed not in Hardy-Weinberg equilibrium. And then let's impose a single round of truly random mating. And to do this, we can use a Punnett square again here, and we can think about the male gametes here and the female gametes in the population here. And these gametes have allele frequencies of the A allele or the G allele as specified here, which we had determined using just on the basis of the genotype frequencies. And now we can ask, okay, if we randomly mate individuals within that population, what are the genotype frequencies going to be in the subsequent generation? And so we just multiply together these probabilities. So we'll have a 0.28 for the AA individuals. We'll have 0.249 for these AG individuals. And we'll have 0.249 of the population will be these AG individuals. And 0.221 will be GG. And so again, in this Punnett square now, we can see that there are two ways to form the heterozygote, as shown here. And so that's, of course, why we add them together. And so the frequency now in the next generation of the AA allele is going to be 0.28. The frequency of the GG genotype is going to be 0.221. And the frequency of the AG heterozygote is going to be the sum of these two, which is equal to 0.498. And so you can see that in a single round of random mating in this population, that the population is now in Hardy-Weinberg equilibrium. And so the answer to this important question here is 1 generation of random mating. And this is important because this reminds us that if we observe that the population is not in Hardy-Weinberg equilibrium, then that tells us that something unusual is going on in that population, that it's deviating from our expectation based on truly random mating. So let's review now some of the assumptions of Hardy-Weinberg's law and some of the principles that have to be in place for a Hardy-Weinberg's law to be acting. So the first is that mating in the population is random, and we saw that indeed that will make a population be in Hardy-Weinberg equilibrium within a single generation of random mating. All genotypes have to have equal viability within the population. The population has to be homogeneous and not divided into subpopulations. And indeed, the population has to be infinitely large. And so these are all simplifying assumptions that are frequently not the case. But these are the assumptions that underlie Hardy-Weinberg's law and Hardy-Weinberg equilibrium and allow us to be able to test a population to see whether something unusual is going on. And so there are some important implications of Hardy-Weinberg's law, as we saw in the example of the carrier testing for cystic fibrosis, is that allele frequencies that are present at low frequencies in the population, in those cases, homozygous individuals will occur with very rarely. For example, if you have an allele that's present at a frequency of 1 in 1,000, so we would say now in this case Q is equal to 0.001, in that case the homozygote for the little a allele occurs at a frequency of Q squared, which is equal to 0.000001, which is 1 in a million. And so that's one of the important implications of Hardy-Weinberg's law. So as I said, Hardy-Weinberg's equilibrium requires random mating, and if in a population it is more likely that matings occur between relatives or phenotypically similar individuals, then this requirement of Hardy-Weinberg's law is violated. And so let's consider some of those violations. So the first important violation of this assumption to be aware of is a phenomenon known as positive assortative mating, or non-random mating. And this occurs when similar individuals, phenotypically similar individuals, tend to mate. So for example, if tall people tend to mate with tall people and short people tend to mate with shorter people, then that violates that underlying assumption, and mating is non-random in the population. And in this case, we would expect to find an excess of homozygotes for tall alleles and homozygotes for short alleles among the short people. The inverse of this is negative assortative mating or disassortative mating, which is when unlike individuals preferentially mate. And this indeed occurs in the human population. And the one idea is that the MHC locus, which is a locus that is involved in the immune system, influences mate choice, as it appears to be that there's a tendency for matings between individuals with dissimilar MHC loci. So you preferentially mate with individuals who have a different allele than you have. And this was famously demonstrated in a study in which individuals, females in this case, were presented with smelly t-shirts that had been worn by individual males. And those t-shirts had been worn for a few days and it got nice and stinky, and they were stuck in a box, each one in an individual box. And the females in this case were asked to come along and smell each of the t-shirts and indicate their preference. And according to this study, they showed a strong preference, and when the researchers looked at the MHC locus, there was a bias towards preferring those sweaty, smelly t-shirts that came from individuals who had a dissimilar MHC locus. This study has since been difficult to replicate, so there's some questions about the validity of these findings. But this principle appears to be true, that in the human genome there are, indeed at the MHC locus and other loci, there is evidence for preferential formation of heterozygous individuals. Another violation of Hardy-Weinberg's law is the phenomenon of isolation by distance. So it's pretty obvious that individuals are more likely to mate with someone close than someone living on the other side of the continent. And this results in this phenomenon of population structure, where locally populations can obey Hardy-Weinberg equilibrium, but globally you might get an excess of homozygotes as a result. And another a related violation of Hardy-Weinberg equilibrium is that this phenomenon of marrying and mating within a population can result in the phenomenon of inbreeding, and inbred individuals are more likely to be homozygous than the progeny of non-inbred matings, and this is something that we're going to talk in depth about in the next lecture. And so this lecture, we've covered Hardy-Weinberg's law and Hardy-Weinberg's equilibrium. Hardy-Weinberg's law, even though it's very simple, it provides an expectation of the relationship between the allele frequencies in the gene pool and the genotype frequencies in the population. And this is an important null hypothesis for us that we can then use, because if we detect an excess of homozygotes or an excess of heterozygotes, this tells us something either about the population or the locus that might be very interesting. And that concludes our lecture on Hardy-Weinberg's law.